Good afternoon, everyone. For those of you in UK time zone, so welcome to the latest of the SCI Fine Chemicals Group and Young Chemist Panel webinar series. So today then, I'll be chairing the session, and I'm also here with my colleagues from the YCP and the FCG, Oliver Ring and Fred Hancock, who are going to be moderating the Q&A session later. So both of these panels are here basically just to facilitate links between organic chemists in industry and academia within the UK, and also provide a forum for discussion and sharing of information relevant to all aspects of pure and applied synthetic chemistry, and that will go beyond the UK, as with today we have this international webinar. So we will have a Q&A session at the end for all of you to present your questions to our speaker. Everybody is in listen-only mode, and you are all muted, as you've probably seen already. So if you need to ask a question, please type a question at any time into the questions box, and that will be available from the start of the broadcast. Just before we do start, I'd really like to thank our sponsors. So AstraZeneca is kindly sponsoring our entire webinar series, which has been going on for 2 months now, and we have further webinars to come. And then particularly for today, we're also very glad to thank ICA for their sponsorship of this individual broadcast. So today I'm really excited to introduce Dr. Kevin Lam from the University of Greenwich. In 2010, he received his PhD in Medicinal and Synthetic Organic Chemistry from the Catholic University of Louvain. His doctoral work explored the use of electrochemistry and photochemistry within the sphere of organic chemistry. He then moved to the University of Vermont, and there he focused on applying analytical and physical electrochemistry with spectroscopy to study the complex redox behavior of organometallic compounds. In 2013, Kevin accepted the position of assistant professor at Nazarbayev, I think that's how you say that, sorry, University in Kazakhstan, before joining the University of Greenwich as a reader in 2017. Today, Kevin will be talking about electrons, cations, and radicals, and electrifying journey. I'll pass over to you, Kevin.
All right, thank you for the invitation. I'll try to turn my webcam on. Good. Okay, so let's go. As I promised you, if you have seen online on Twitter or on LinkedIn, today I'm not going to give you the usual fully synthetic organic lecture or presentation about what we have done. I will try something a bit more exotic. I'll give you an overview, of course, of organic electrochemistry, but also on the side projects that we are working on that have kind of nothing really to do with it. But at the end, I promise you, everything will start somehow to make sense, and I'll give you probably a broader view of the field. So let's go, and let's start with the first question. Whenever you actually mention the word electrochemistry to an audience, it literally depends to whom you're talking to. I mean, literally, you will get these cliches. My friends honestly think that I'm making batteries. Not really. The students hate it because it's full of equations. Most people think, thanks to ICA, that all I do is to play with nice toys. My colleagues are always wondering where I am, and they really think I'm only hanging out with Phil Barron. Yeah, I mean, the guy here is actually Phil Barron. You can see that we're not going to the same gym, apparently. But the reality is more what you can see on the last picture. It's something full of wires that looks complicated, but really, when you look at it a bit closer, it's not going to be that complicated. So I'll try to take you onto a journey, literally through the fields, and the main theme will be electrochemistry, and you will see how we will start with analytical organometallics, and we will somehow end up with medicinal chemistry. So we'll start with that. This is usually something that organic chemists, I wouldn't say are afraid of, but starts to hate. It starts to be too analytical, too many equations, fine. But as I said, I will try to convince you that there's a link between that and the development of potentially a new drug. So let's go. And as I said, for the whole talk, you will have to to be open-minded. It's going to be a bit exotic at some point, but again, I think it's giving you a better view of really what modern electrochemistry is, rather than these old-fashioned just equation types of electrochemistry. Okay, so before we get there, we have to agree on some concepts. And one issue is that normally, whenever you talk about a reduction, and here you have an example, reduction of a ketone, people from different subfields are going to understand something very different. So this is the difference, the dichotomy between the organic chemist and the electrochemist. So let's look at the organic chemist first. For an organic chemist, this is a reduction. So this is the reduction of acetone with lithium aluminum hydride. And you see a mechanism that you all know, nothing really exotic here. But pay attention to one thing. Really, you do not have a single electron transfer. This is what an electrochemist will call a chemical mechanism, meaning that you're going to transfer acetone. You're never transferring really a single electron. On the other hand, if now you ask an electrochemist what is the reduction of a ketone, they will come up with such an equation. The monoelectronic reduction of ketones is what they will call an EC-EC mechanism, meaning an electron transfer followed by a chemical reaction, followed by another electron transfer, etc. I always joke, but it's fairly easy to be an electrochemist. You have to be able to count up to 1, because we think as a single electron transfer. So one by one, we never ever really transfer 2 electrons at the same time. So if we agree with that, we can move to the next small issue, electrosynthesis. The same word doesn't mean the same thing between the two fields, or the views are very different. For an organic chemist, you buy your electrosyn, and this is what you do. You just put things in the cells, you start the electrolysis, and eventually you get a product some way, and you record maybe cyclic voltammograms, and you say, oh, that curve, I don't really like it because it's not perfectly reversible. I like that one because it looks like a duck. Now, for an electrochemist, this is, the duck-shaped curve is really dull because it means that nothing is happening. It's very nice to look at, but in terms of synthesis, it's not very exciting. On the other hand, if you do the same as an electrochemist, you get that. You get a lot of equations, you get these equivalent circuit diagrams, but then they have no shame, because if you talk about, let's say, reduction of cyclohexanone, they have literally no shame to tell you that, sorry, the oxidation here, that the oxidation, can lead to such cation and radical. They will break bonds where normally you should not be doing that. So it's a bit controversial also when you look at electrochemical mechanism here. So for the rest of the talk, I'm going to try to stay in the middle, because we will have to understand both fields, and I will give you a good overview of what it is to be an electrosynthetic chemist. So in this talk, you will see I'm going to link all these topics together. So surface catalysis, meaning we can actually put a catalyst on a surface and do chemistry with it. Synthesis, that's the big chunk of the talk. I will talk about organic reactions using electrochemistry. Mechanism studies, a little bit about organometallic here, and eventually at the end of the journey we'll reach what, I don't know, I don't have really a name for that field. I call it medicinal synthetic electrochemistry, but eventually we'll get there, and this is still a working topic, but it's quite exciting, at least to me, and we'll see why in a bit. Now, what are the big positive points of doing electrosynthesis? Probably the first one is that you will have a hard time to find a reagent which is cheaper than the electron itself. This is coming from the current, the electricity. It's extremely cheap. It is most of the time quite safe, or much safer than normal synthesis usually. It's green, you can argue, depends on the solvent you use, but you use a renewable source of energy here. But also I will focus on reactions that are really unique to electrosynthesis, and most of them that I'm going to show and talk about today could not be performed in a normal round bottom flask with chemical reagents. Okay, so we are going to start as far as I can from medicinal chemistry by looking at a very simple compound, which is Ruthenocene. So everything starts with Ruthenocene. If you look in the literature, you might think that Ruthenocene has been studied to death. And indeed, the oxidation of Ruthenocene has been described in hundreds of papers, and they will always tell you, you cannot oxidize ruthenium-2 to ruthenium-3. You always end up with a ruthenium-4. Okay, that was an accepted fact. However, this is due to something that most organic chemists and even organometallic chemists are not looking at. Whenever you do electrochemistry, you need to have a salt in the solvent for the cell to be conductive. And it happens that whenever you have these anions here that are quite strong coordinating, actually, although organic chemists call them weakly coordinating, it really depends what you have as a cation. These are actually quite reactive. So they actually form these ruthenium-4 compounds here. So we used a little trick. Okay, you might argue it's a bit of an expensive trick, but if you use these very large anions that are much weaker at coordinating, we've been actually able to observe the formation of a ruthenium-3. I'm not going to go through the curves here in details. I'm just going to extract the important message is that if you do that, you can now observe the radical cation. It's a 17 electron complex of ruthenium. The thing about it, as a radical cation on any organic molecule, if you have a radical cation, two of them can dimerize, and you get a dimer. Here's a metallic dimer, but you can get a ruthenium-ruthenium bond, and eventually you can reduce it back also to the monomer. And you will see why this is important. So we've done that with open sandwiches complex, the Ruthenocenophane here. They dimerize it in the same way. You can even crystallize them. You can get the X-ray, fine NMR. What we were interesting as good electrochemists is once you found something like that, you're just going to apply it to every single compound you can find. So we said, okay, move on. And let's try to do that on rhenium. Same behavior. You can oxidize rhenium to rhenium 2 here that will dimerize also, and you get the same type of dimer. Fine, not a big deal. What we were interested, and you will see this is going to be very important for the rest of the talk, is in the possibility to exchange these CO ligands by electrochemistry on these compounds. It is known that you can just do photochemistry with these CP carbonyl compounds and exchange a ligand, but it's very sluggish. If you do it, yields are very low, and it's really, not the best way to perform that reaction. On the other hand, you can use electrochemistry, and this is what we wanted to do. Generate the radical cation. There you can easily exchange a ligand and then reduce the compound back to the substituted compound. So this is what we wanted to do. We took the rhenium and a cyclooctene, and yeah, nothing happened. And this is an example where you need to be a synthetic electrochemist to actually take to actually take 5 minutes to look at these curves. If you look at them, the only thing that is different when you increase the amount of cyclooctene is that little blip here that is increasing. If you are a pure electrochemist, you're going to say, well, it's just the cyclooctene that's not pure. And that's why, this bloody organic chemist gave me something that's not pure, and I see like a little impurity maybe here. If you are an organic chemist, you just look at this curve and you say, bah, they're all the same, nothing happens. Now, if you are someone like me in between the two fields, you look at that and you say, hey, there's a catalytic reaction. Something is happening. And indeed, this is when we started really to look at organic electrochemistry. If you use rhenium 2 as a catalytic oxidant, you are able to start to trigger a catalytic 2+2 cycle addition of non-activated olefin. They make cyclobutanes here, fine. You just need a very small amount of rhenium, 5 more%, even 1 more% works. Be careful, you cannot use these anions. You really had to use the big borate. Now, this is okay. I mean, this is a pretty, I mean, I'll be honest with you, this is really a useless reaction because it only works on completely non-substituted olefin. Like if you have any functional groups, the whole chain doesn't work anymore. So that was fun. I mean, we made these cyclobutane, not a big deal. But that was a concept really here that I wanted to show you, that you really have to be open-minded and do not discard a result that do not seem perfect, because maybe there's something behind that. And at that point, we decided, okay, let's do some organic electrochemistry. So this is our first, as I call it, sightseeing stop, the organic chemistry that we do using electrosynthesis. I always joke that if you ever attended a webinar by Phil Barron and all the big names, they will show you like the latest trend in electrosynthesis, while I will do completely the opposite. I'm going to talk about a reaction that has been discovered in 1800s, a little bit earlier actually than that, by Michael Faraday himself. So this is going to be extremely old chemistry, but still I will show you that there's some merit in it. So Michael Faraday and a little bit later Hermann Kolb have discovered that reaction. If you take 2 carboxylic acids and you oxidize them in a cell, you get a dimer here from the alkyl part of the carboxylic acid. And the question is kind of puzzling, but how do you manage to get that dimer? So here you have a little scheme, and I'll show you the first interaction here. It's going to be the adsorption of the carboxylate onto the electrodes. Then you have an oxidation. You form that a radical, which is absolutely unstable. That will decarboxylate. You form CO2 that is leaving your cell, and you form the first radical coming from the carboxylic acid. Now, the beauty of electrochemistry is that it doesn't really matter what happens here in the bulk of the solution. What matters is what is happening next to the electrode. So imagine, I'm going now to put an excess of a second coacid, and if you manage your system properly, you will be able to get a huge amount of these radicals in very high concentration next to your electrode here. And what will happen is that you're going to recombine them to form here an heterodimer. Again, it's interesting that already in 1800s, it was actually reported, probably the first cross-coupling that is not even catalyzed. This is a metal-free, catalyst-free sp3 cross-coupling starting with non-activated really compounds. Okay, so practically, of course, you can have your own setup that you can build by yourself, but practically what we do, we use these electrosynths, and you see like they have options here to run parallel electrolysis. So this is our go-to system to run these reactions. So here, this is a little bit of product placement. You can see like Matt and Alicia quite happy holding the electrosynth. We looked at that same reaction, the Kolb reaction, but probably in a bit of different ways. We have published before, and I'm not going to get into details here, a way to actually generate radicals. I mean, these carboxyloxy radicals that could be trapped here to form lactones before the carboxylation. But recently, we became interested in another way to form lactones, which I always call it the silly way, because this is not what you learn in unis or in textbook. If you want to make the lactone, for instance, normal people would do the disconnection in red here. what we thought is, hey, why not do that blue disconnection here, which is normally something you don't do because it's not that easy. How are you going to form a syntone here? And we thought about it, but it was fairly easy for us. We said, okay, we need that radical. And if that radical is coming from a decarboxylation, it's simply an hemioxalate ester here, which you can make from the corresponding alcohol and an excess of oxalic chloride. That's one step. Then we decided, okay, let's push the system a little bit. If we take these precursors here and we have a coacid, can we actually form these functionalized lactones here? So the idea is we form the corresponding radical, that one cyclized under the in saturation here. You form a new carbon center radical, but at your electrode, the coacid that you added in slight excess will decarboxylate to form a new radical, and these two will recombine with each other. So we did it, and this is, I always call it the dreadful scheme with all the results, but I haven't found a better way to present them yet. Again, I'm not going to go through each of them. I'm just going to show you that, yes, you can use coacids here that are quite bulky. It's fine. You can use functionalized coacid with an ester. That hasn't been an issue either. You can have, interestingly, chloride, bromide here. Keep in mind that normally halides are not always well tolerated whenever you do radical chemistry. Aromatic compounds are fine, secondary coacids are fine, and even a CF3 is fine coming from the trifluoroacetic acid. Very, very cheap source of CF3 radical, by the way. So we did that, and then what we did was to do the experiment to many. You know, if you have ever seen Frank Glorious, he's telling you that once you have a working system, try to do some perturbation, temperature, water and everything. So temperature on the reaction doesn't do much. Then I asked Alessia, can you actually use wet solvent? No problem, the reaction was still going. Can you add 1%, 2%, 3% water? Was still going, till the point that she did the reaction on or in water and it was still going. So basically we had to relook at some of these results to say, like, can we actually do the reaction in 100% water? And the answer is yes, you can do it, but your starting materials have to be soluble in it. So that kind of a small limitation, but you can use water if you want. That also means that you can do these reactions without having to dry your solvent, deoxygenate. This is radical reactions for almost dummies, I said, like it's 101. Just take the solvent out of your bottle, mix it in the cell, electrolyze, go get a coffee, come back, and your product is there. In the same vein, we also looked at the formation of what is called nootropics. Apparently, these compounds are supposed to make you like clever or make you think faster. I don't know if it's true or not, but they are an important class of compounds. So we made them following the same strategy, generating an acyl radical here that could cyclize here to make these lactame. And once we had that, we can recombine with the coacid. Again, I'm not going to go through all of these results, but you get the idea. You can do radical cyclization and trap the radical center, the carbon center radical with another. So, okay, we've done that. More recently, we also look at the MOM chloride problem. You probably know that MOM is a classic protecting group for alcohols. And yes, it's come with a drawback that using MOM chloride is not fun because it's quite toxic. So we decided to look at an alternative here, which is the chloroacetic acid. And that will lead eventually to the same MOM ether. Now, in the whole sequence here, it's quite nice because there's not a single purification. You do a Williamson etherification, you get that ether acid here, electrolyze it in methanol, you get the corresponding oxonium, methanol traps it, you get your MOM ether, simple basic workup, no purification, solvent right out of the bottle, and we made these examples. Again, I'm not going to bore you with every single one of them. Just remember that it's simply working, okay? You can make grams of if you want, of like MOM ether here without any issue, really. But of course, there are compounds that are not working, and usually we don't talk about them. But any compound that has, for instance, a benzylic group cannot be eterified like that, simply because the benzylic position is actually getting over-oxidized over the time. So the idea is, to solve that, what you want is to have the compound into your cell, transform into the MOM ether, and then your compound out. You do not want the compound to stay in the cell to get over-oxidized. And the answer to that is, we need a flow system. So I kindly ask Alessia and Matt to try to find one. And then eventually we did find it, but then we found also the price. This is really expensive. And also it takes a lot of space. So I'm not going to lie, we cannot really afford it. So if anyone in the audience wanted to sponsor us and buy us a real flow system, we're more than welcome. But I've learned something during my PhD and postdocs. If you cannot get something because it's too expensive, try to ask or try to make it. So I just went on Twitter, twisted a little bit the whole demand. I said, okay, can someone 3D print me a small item? And then Adrian Dove, my head of department, said, hey, just ask my friend Steve Hilton, who dramatically said yes. So here it is, the very happy Steve Hilton, probably before he knew what I wanted. But yes, we made something which is 3D printed, which is an electrochemical flow cell. So the electrochemical flow cell is a bit of a MacGyver thing, but you can see here we have like 2 electrodes, and between the 2 electrodes we have a flow path here. So your solvent is just going, to go to flow here, and it doesn't take a lot of space. It's very small. You can print like 10 of them for like less than £5. It's really, really cheap. You can also connect it, of course, to any power supply. But because Steve Hilton is a bit of a perfectionist, he also built the entire setup here where you can now use compressed air or compressed gas instead of a pump, because the pump costs a lot for this system. But here you see that all we're going to use is compressed gas to pressurize a bottle. The solvent will come out and it will then flow into the cell. Of course, if you know Steve Hilton, he wants something that looks nice. So he made the entire setup that can actually fit the Electrosyn. You can literally click that little flow cell onto your Electrosyn and transform your Electrosyn from a batch cell to a flow cell. And I call that, this is not an Electroflow because the trade name is already taken. If you Google Electroflow, you will find a boiler or something like that. I have no clue how we're going to call it, not a big deal, but this is completely 3D printed, okay? So everyone can have an not an Electroflow at home or in the lab easily. Then the reaction is now quite easy to follow. We take the potassium carboxylate of our substrate, we put it in the cell. Once methanol is flowing through the cell, you have no current. Once your compound gets into the cell, you get a current, so you see it. And once the MOM ether is out, you have no more current anymore. So easy. Again, we got okay results. I'm not going to lie, the flow didn't really improve much the yield for any of the one that worked in batch. The only one which is really, really interesting is now the one compound that is bearing a benzyl group here. It is now giving really good yield in flow, while in batch we couldn't get any of that. So that's probably the merit of batch compared to flow in our case. So this is, again, just a summary of what we have done. And I always put that it actually does work. It looks like a toy. It's 3D printed. Anyone can have it, but it does work. Trust me, it's a nice little toy. And before we leave organic chemistry, this is probably just because I'm coming from a lab that has dealt a lot with Orthoesters. This is a topic that I really love, is how do you make these compounds, the Orthoesters? And here I will show you that we've developed a new syntone, very easy, to generate almost every single possible autoester in the world that you want. But maybe before, the question is, what are you doing with autoesters? Well, first reaction, very easy. If you mix an autoester with a diamino or an amino compound, you get benzimidazole, benzoxazoles, and benzotiazoles, everything you want, fairly easily, no metal, just a little bit of HFib here. Stir it, and you get very good yields in these fragments here. You can make amides also in their very mild conditions. You can use them as activated electrophiles, equivalent of, if you want, an oxonium of an ester, and you end up with, actually here, if you see, like, it's kind of an aldol reaction, but you end up with a protected ketone and a non-protected ketone, which is fairly difficult to do normally. You can do the classic Claisen, Johnson-Claisen rearrangement. Sometimes it's also used to specifically esterified a tertiary alcohol in presence of primary when you can go through these intermediates. So they have use, but I do think they have been underused, because once you look at how you're supposed to be making them, it's a nightmare. The go-to method, really, that everyone is using to make them is called the Piner reaction. You have to start from a nitrile that usually you make with cyanide, not great. Then you have to use HCl gas, sometimes HCN gas too, to form these amidate salt here. And then you have to decompose it with an alcohol in very dry conditions to avoid the hydrolysis. And it can take up to 6 days, and yields are not fantastic. They're like 40%, 60 in best case scenario. It's just a pain. You can tell why no one is using them really in everyday chemistry. Well, we have found that, hey, you can get them in a couple of hours if you want to. Just take what we call the DTC carboxylic acid here. You make it from glyoxylic acid, kilos if you want. You can get also these compounds by carboxylation of the corresponding lithiodiethyene, or by directly protecting the ketone on alpha ketoester. Anyway, there are many ways to get there, okay? And here's another dreadful slide. Here, really, the take-home message, again, is we made it. I might be wrong, but until today, this is probably the only method available that will allow you to make functionalized autoesters. Because remember, with a Piner reaction, you need to use HCl. If you use HCl gas, not a lot of functional groups will be tolerated. While here, you can see that we have nitrile, we have halides, we have whatever you want. Triple bonds, it's fine. It works on aromatic compounds, works on tertiary compounds, it works on benzylic compounds. Not a huge issue. We can even make diautoesters. And also, we made for the first time a new class of compounds, which are the trifluorinated autoesters that are completely unknown. And now this is a little bit probably the inorganic chemist in me. I don't know what to do with them. They're extremely reactive. I don't know yet what to do with them in terms of synthesis, but maybe someone will find them useful whenever you need a very reactive autoester. Again, you can condense them to make these compounds. You can make it in situ in the cell. You don't even have to isolate the autoester. You don't have to purify. And to show that you literally do not have to worry about purifying, so there's no purification here. You can use the crude and directly engage it into a Suzuki coupling. It works with the autoester still being alive at the end. Okay, so I still have a little bit of time, and this is really now where we're going to start to get into the exotic chemistry. So normally I'm not really giving talks about that bit that often, because this is something we do on the side, but this is the fun thing. So you have seen where I'm coming from: analytical electrochemistry, organic electrochemistry, and now what we're going to do is to put everything back together. And this is the light version. CO2. Can we do anything with CO2? Because CO2 is annoying, it's there. So basically, if you can do something, we have too much of it, it's fantastic. Also, now I put my hat of surface chemist. If I developed a catalyst that can reduce CO2, why don't I actually put it on the electrode so that I don't have to bother to filter it? It's not going to contaminate my product. I mean, people are making supported catalyst. I'm going to make an electrode supported catalyst. And we had to develop a way to put these catalysts on it. And this is a bit of an exotic way, but I love it because it's very easy. Take any alkyne, add a little bit of butyl lithium, make the anion, oxidize it. You're going to make these extremely exotic alkynyl radicals. And again, if you're an organic chemist, you won't think about it. But if you're an electrochemist, you don't care that it's normally not that easy to make. You see a radical, you see a carbon electrode, you're like, it's going to graft on it. And indeed, you can physically, covalently graft anything that has a triple bond onto an electrode. And you see that what is happening is that your graft catalyst here is able to communicate with the solution. So you can transfer electrons into the solution. So that's great. And you see it again, I'm not going to get into details, but that wave here that is symmetrical is for the ferrocene that's on the electrode. And that one is for the ferrocene that is in solution. So you can see that they talk to each other, they see the existence of each other, and they work together. And this is exactly what we wanted. Now, I'm not going to keep the suspense too long. We haven't done it with the real catalyst yet. We're not at that point, but at least we are ready. So we thought, okay, with our colleagues in the US, we said, we need to develop some ways to reduce CO2, because basically this is what is paying the bill. Everyone wants to reduce CO2. And okay, so here's the very simplest idea. Take a metal. This is a Lewis acid. Your CO2 can bind the Lewis acid through the oxygen. And then we'll have a donor, and the donor will attack the CO2, now the activated carbon here. And what happens is that you kind of bend the CO2. And if, like me, you were not a CO2 specialist, I have to tell you that pre-bending the CO2 is making it much more easier to reduce than the linear CO2. Now get ready to be amazed by the very complex catalyst we helped to develop. It's that one. So this is literally flipping through the catalog of Sigma Aldrich. You can buy that pyridine here. It doesn't cost that much. You can buy tons of zinc chloride. And then what we have done, and we have replicated that in US and in the UK, you just get a high school student and you ask them to mix both together, and you get the very complex catalyst here. If you do infrared, you can see that it's actually that wave here, that band here is for complex CO2, so it's there. Now, the videos are going to be a bit choppy. I'm very sorry about that. But what I want to show you is that here's a solution of the complex, okay? If you bubble CO2, you will see a white solid precipitating, which is the complex adduct. Now, let me be overly honest with you. Transforming CO2 is not a challenge. If you look through the literature, everyone is transforming CO2. But being able to trap CO2 from the air, and we cheat a little bit here, you see that we use coke, okay? It's a bigger challenge. So here you have coke that is being poured into, they have done that in US, into a flask. And then we're simply going to stir. So you don't have a high pressure of CO2. It's wet. Who knows what's coming with it? I mean, it's not a refined pure CO2 coming out of your cylinder. We plug it into the small vial, and you will see it's going to become turbid here. You can see that it's not as clear as before, showing that the catalyst is actually trapping the CO2. I'm not going again to go through these CVs here, but one CV is able to tell you that the turnover frequency of your complex is 96 second minus 1. It's very slow, but the great thing is that it gives you selectively here CO. So it's reducing CO2 into CO. And usually, I always joke, because we have transformed the greenhouse effect gas into a deadly toxic gas. So kind of move the problem to something even worse. But what we want to do is here on this slide, we want to 3D print eventually a system that can generate CO safely through the reduction of CO2, and then eventually use it in maybe cross-coupling here, or use even radio-labeled CO2, probably CO11 to be honest, to do PET tracers. It's a bit of a dream yet at the moment, but we're working on it. We maybe one day we're getting there. Okay, and now here's the final topic. I hope I still have a little bit of time. This is the topic. Usually, I rarely talk about this because usually no one fully understands how I got there. But at least now, all the listeners of the SCI have kind of an idea of my background. And this is where, basically, whenever I give that talk to graduate students, this is basically every single thing in one topic that you should never ever do when you think about research. So let's go. First of all, whenever you design a drug, should you take a metals? And if you ask people, they will tell you that you're crazy. Do not take a metal in your drug. Why? That guy called the Smurf, and I'm not making that up, it's really how he called himself, decided to take silver every day in his life, died at 60, I think. Not a great idea. Cadmium in Japan poisoned the river and people. Is attacking your bones. It's known. Mercury goes into bones, brain issues too. Basically the Mad Hatter. It's called the Mad Hatter because people were using actually mercury in felts for hands. Oops, sorry. Arsenic, quite toxic, as they call it. Arsenic, the poison of kings, the king of the poison. But when you look in the literature, it's not fully true. There's something called Trisenox, which is arsenic trioxide, and this is sold as a drug against leukemia. Also, if you know a little bit about leishmania, there's an antimony, actually, drug that is used against leishmania, and you can see it is actually working. I mean, I'm sorry for the pictures, but I think you get the point. After treatment with antimony, you do get rid of the parasite. So, okay, really the take-home message again is it really depends on how much of a metal you're going to take. So I told my colleague at that point, the real medchem one, because I'm not a medicinal chemist, I'm just pretending, I have a great idea. I'm going to take back, much before COVID, the infamous chloroquine, and I'm going to add a metal in it, and I'm going to decorate the thing a little bit to make the whole thing stable. And they looked at me and they said, well, okay, because someone actually made ferroquine with ferrocene here, and I said, I can make something. And they looked at me, the med chemist, I said, yeah, sure, go play. I mean, you're an electrochemist, organic chemist sometimes, go play in your lab, sure, do it. And this is normally where you decide, okay, should I really do that? And because I'm quite stubborn and do not always listen to wise advisors, I decided, yeah, let's do it. And we decided to target actually melanoma because actually chloroquine was shown in clinical trials to be efficient against melanoma, not fantastically efficient, but it has shown to have some effect by inhibiting some of the BRAF here, but mostly a mutation. And also you'll see later, it's fairly easy whenever you have mice to actually measure the progression of a cancer. The way chloroquine is targeting the cancer is by actually stopping the autophagy in cancer, by literally destroying the lysosomes. As I said, chloroquine is already in clinical trials, so we know that we knew it was probably a good starting point because it's already FDA approved and it's not fantastically, not fantastic drugs, but we thought, okay, maybe we can do something with it. You have heard of chloroquine as maybe now with COVID, because what I thought, and maybe it's working against COVID, I'm not quite sure, but it does come with a lot of side effects. It can have, I mean, it can lead to seizures, skin rashes, eyes issues, etc. And it doesn't work very well in an acidic medium, which is a problem because usually cancerous cells are a little bit more acidic than other cells. So the way chloroquine has been described, the way it works is that the chloroquine gets into the lysosome, gets protonated. Here you increase the pH of your lysosome, which is basically going to neutralize it and block the autophagy. And once it's protonated, it cannot come out of the lysosome. Okay, so I said, fine, let's put my favorite metal, manganese tricarbonyl here. And this is where I got a bit of a strange look from my colleague, because if you ever do Google manganese, intoxication with manganese, you will find that it's quite neurotoxic, and there's something called the Parkinsonism due to manganese. So you can see like the Paul Albert Hoffman here being exposed to manganese intoxication, and you see that eventually his handwriting becomes a little bit shaky, I'd say. And then really to make sure that I put every single problems in my equation, it comes with CO ligands, and CO is highly toxic. But I was like, doesn't matter, you can get tons of these compounds because it has been used as an anti-knock agent. Great. So here's what we did. We basically made something that we called cimenquine, because this is called cimentrine. I'm not going to get into details for the synthesis. There's nothing really creative here. It's linear, but as I've been told by people in medchem, doesn't matter. All we want is the final compound. So we made it. Not always easy, but we made it. We have no clue how it works, because if you look at the 3D structures of it, it's fairly different. You have a huge manganese atom now in the drug, very different in 3 dimensions. And of course, if I'm giving a talk, it's probably that we got positive result. But this is a message for graduate students and undergraduate students who might be with us today. In real life, what you don't see behind the talks is that it's quite something like that, okay? But let's be positive, and I will show you the result here. So my colleagues in biochemistry did these experiments. They tried the drugs on human melanoma cells, and you see that at almost neutral pH, our drug is outperforming the chloroquine. There's no picture. We do outperform the chloroquine. If we mimic the acidic environment of a cancerous cell, you see that yes, our drug here is losing in efficacy. However, chloroquine is almost not active at this point, because probably it gets protonated even before it can get into the lysosome. They try that also on different cells line, and maybe if we look at the kidney cancer here, you see that chloroquine under normal pH has an IC50 of 26 micromolar. It's not fantastic, I know. Cimenquine 7, still micromolar. People are not happy with that, but we have huge issue of solubility. But what is interesting is that if you now move to an acidic medium, you see that there's a huge drop of activity for the chloroquine, while cimenquine is still active. It hasn't completely been inactivated by the acidic medium. This is just a summary. Basically, the metallic drug works better than the fully organic drug. Again, we're not fully sure how this is working. One experiment they did to help us was to really make sure that it does target the autophagy machinery here. You can see that they've targeted with the fluorescent protein. And when you use 10 micromolar of cimenquine, it's almost completely gone. Well, chloroquine, it's still there. The cancer can still do autophagy. Okay. Then we come back to the first or second slide I've shown you, because this is a tricarbonyl compound. So what we have done, we just used analytical electrochemistry, and we changed the ligands. So we made a new type of libraries for medchem. We're not changing the organic ligand. We now change the redox potential, the solubility, and all the possible physical properties of the metallic center. And we haven't tested them, all of them yet. But my guess is that we'll find that the bioactivity is linked to the redox potential of the drug. So there's a little bit of still suspense here. I don't know. But this is like tying up basically all our experience in organic chemistry, organometallic chemistry, to apply it to a real life example, which is a drug design. We are also going to do something quite exotic. So because my colleagues in the US didn't really follow all the rules. They decided that yes, we can try it on mice. And we tried on mice. None of the mice died, which is really good. So it means that the drug is not too toxic. You see that it's working. It has an effect against skin cancer because you can just measure the growth of skin cancer. It's not fantastically more active than hydroxychloroquine, but it is working. And to me, that's enough. I'm already very happy that this very weird exotic idea is actually working. It's doing something and it's not killing the mice. Now the mechanism, we have another idea, which is again going to combine biochemistry, spectroscopy, and inorganic chemistry. You might not know, but this is how physicists and most inorganic chemists are looking at the infrared, upside down compared to organic chemists. These CO bonds have extremely strong stretching, 1,000 times stronger than normal carbonyl. So we can see them even in aqueous solution, even in organic medium. So now what we are going to do is to inject either on cells or either on mice, draw some sample, and we're going to use actually infrared microscopy to be able to locate the drug within the cells. Not only locate the drug, but also being able to quantify it, to try to understand where is the drug going and how does it work actually, and how does it cross all these membranes, although it has a bigger atom. Just before we finish, there's a point where you shouldn't be ashamed to rebrand, like Apple, okay? You see that they rebrand themselves all the time. It happens that Cymanquine is working against malaria with a much better activity. We are now in the nanomolar range. And if you compare Cymanquine here to artemisinin, it's not that far off. I mean, of course, there's quite a bit of an error here, but it's not too bad. So maybe it's maybe an interesting anti-cancer compound, but it's pretty much very interesting also against malaria, because if you remember, artemisinin and TUDU actually won them a Nobel Prize. So there's kind of an interest to develop new anti-malaria agents. So we're working on it. It's, as I said, it's very slow for us because it's a side project. It's not the main thing that we do. But this is where I really get excited by all these ideas. So thank you for having been with me and bearing with me. I warn you that would literally be a journey, because for once I haven't focused only in the organic chemistry field. And I really think that nowadays as chemists, we have to diversify ourselves and see where we can work together. But of course, I hope I've convinced you that electrochemistry is a fantastic tool that is still underappreciated, in my own opinion. It's cheap, it's green, and I've shown you some unique reactions. Of course, I haven't been the one doing all these experiments alone in the lab. These are the people who are helping me. You see also that I call them the real electrostars. We are a very small lab, but we are having a lot of fun. And I do think you don't always have to do research in academia because it will lead to a JAX, it will lead to a science. You do have to do things because they're just fun and interesting. And this is what is driving us. All these collaborations became friendships, and working with people that can bring you new ideas and try to collaborate with fields where you have to learn a lot because it's not your own field. It is really exciting, at least to me it is. And the last slide before I finish, you might know her. Apparently she's quite famous. This is the infamous Catherine Wheelhouse from GSK. So Catherine and I have accepted to co-edit an edition of OPRD coming next year sometimes about electrosynthesis. So if anyone in the audience is interested about sending a paper to that special edition, please get in touch with Catherine, or please get in touch with me if you want. The deadline is in February, okay? If not, it will be open also online, but we will really welcome anyone if you have anything to publish there. Well, thank you for your attention. Thank you for bearing with me. And I think we might have some questions now.
Is it okay? I've got Kevin. I've got one question that came up that's come up on the flow of electrochemistry,
Yeah.
and that's from Chris Brown in Evotech, and he's asking about. He was asking about the consumption of the electrode in the flow electrochemistry experiments. Was there any significant consumption of the electrodes?
If you have carbon, yes, you will have. I mean, carbon electrodes are merely powder that have been agglomerated. It's a bit like this cheap IKEA thing that you buy. Eventually, at one point, they will put it in the same way in powders that are going to there. So with carbon electrodes, it's true, but because it's so cheap to make, we use them as consumables. Basically, you can just dispose them. We have made some metallic ones. Or if you use actually metallic ones in any flow system, these ones are not getting consumed at all. So you'll find it really depends on your application. With carbon ones, yes, it's going to corrode at one point. And in our case, it's cheap and readily available, so we just get new ones.
Okay, so we got a question from Paul Hernandez from Oxford. It's also a question of mine as well. So he says, do you have any data regarding cell penetration of chloroquine versus your modified?
No. When you do not know, it's okay to say no. No, honestly, no. We have, as I said, we have no clue how it's working. It's serendipity. That's it. We're working on it. Yeah.
Yeah. So the question goes on to say, you mentioned about protonation, but in terms of PKs, you would expect them to be pretty much the same. So
it's not.
What modification do you think causes that, or is it just?
I mean, it is not in terms of PK. I mean, we measured it. I forgot to be honest what the real PK. It's not as basic, and so we try to understand why, because it's not directly bound to the CpRe ring of the metal. So we're like, well, could it have such a big influence on the whole PK? It has, but we still do not understand how. So I gave that challenge to some of my physical chemistry colleagues, and they're really swearing at me because apparently having like heavy metals in calculations is not something great, because whatever computer and power you need more, it's going to be longer. Honestly, I don't know. This is why we decided to use spectroscopy to follow the drug, to try to understand really where it's going. Because the drug has a fantastic infrared tag also. What we know is that it's going into the lysosome, because I know that my colleague in biochemistry have taken samples of the lysosome solution and did HPLC-MS, and they've been able to quantify the drug in the lysosome, and it's mainly going there. Again, why and how, it's a mystery. If anyone has a clue, I'm happy to take it, because we don't know.
So clearly having the metal in there is positive. Have you ever switched it out for something else? Switch it out for a different metal?
Not yet, because two things. One, the academic point of view is the price. The next metal in my list, which could probably do the job, is the CpRe rhenium. Rhenium has a very different price compared to manganese. And yes, eventually, you know, when we get it funded a bit more, we might. Also, you are limited by one very important factor in these drugs, is that, I know it might sound silly, but they have to be stable to be able to inject it into water, and not every single organometallic are. So in terms of, I mean, I'm interested in metal tricarbonyl complexes. There are not that many that are so stable. So we decided to use the manganese first. The rhenium, we'll use it. It's in the pipeline. Once we are good with the manganese, we'll switch to the rhenium. I know they try, not us, other people try the Ruthenocene. Doesn't work as well.
Cool. Fred, do you want to go next?
Yeah, there's from Thomas Gutovas, and he's asking about using electrosynthesis for supramolecular reactions?
Yes, you can. You can. It is not unknown. It's not, I think, as easy, because keep in mind that if you want to use it for supramolecular chemistry, you do have to be able to get your polymer or any supramolecular entities solubilized into solvent, because electrochemistry can only happen in solution. Anything that's not soluble will not get electrolyzed. But it has been used. I know people have studied like for polymerization, but also functionalization. It's just a little bit more challenging than on small molecule, mostly because you need to think how the absorption will happen and also how to analyze your product. But this is, I mean, something you know better than I do. But yes, no, it hasn't been used that much compared to the other fields, but it's possible.
Okay, we've got a question from Kim Rupa. Sorry. Hi, Kim. So you said that you think that the SAR of your compound against cancer will be changed with different ligands, and so the redox properties of the the redox properties of the compound. Does this hint at all towards the mechanism of action? How are you planning on studying this?
Honestly, we haven't done. I mean, I'm going to tell you, but we haven't done like intensive bio testing on these new compounds. But from a previous experience, we can actually tune the redox potential of the drug to get like whatever you want, basically, by switching with the one of the ligand with the appropriate other ligand, 1 or 2. You can tune it. You can have like 1 volt versus ferrocene, 1.2 volt, etc. And if you see actually that there is a correlation between the bioactivity and the redox potential, it probably means that what you have is a pre-drug and not the active form of the drug. What happens is probably that the ligand get oxidized to another cytotoxic form, because once you are out of the proper redox potential, that oxidation doesn't happen, or reduction, but usually it's an oxidation, doesn't happen anymore, and you do not generate a lot of the active species. Yeah, we can easily measure the redox potential of the drugs, and then we correlate that with the bio result we get back. And yeah, another thing that I haven't talked about is these metals can do also Fenton reaction. So it is known, apparently, it's not coming from me, that cancerous cells have a slightly higher amount of hydrogen peroxide. They might be able, if they have the right potential, to actually generate hydroxyl peroxide that will kill the cell. So it might not be a single way of killing these cells. There might be 2 or 3 different ways, but yeah, this requires like EPL studies, which is another tricky thing to do.
Yeah, it's quite a clever way of killing the cell, actually. Thank you. Fred?
Yeah, we have a question, Kevin, from Musa Sahalia, no affiliation. He's asking about the conversion of CO2 to CO, and he was wondering whether you see the intermediate formic acid or something like that would be formed at all.
That's a good question, because you know when you give a talk, you always not lie, but you hide the annoying bit. No, this case, no. It seems to be only giving CO2, CO, sorry. But then we do get usually the annoying question, but okay, where's the missing oxygen going? Because, you know, I mean, once I was giving a talk somewhere and I've been told that I don't know how to balance my equations, or like, thank you. The thing is, there's always, although we do that in a growth box, I mean, it's very difficult to get complete setup completely dry without like 1 ppm of water. And I've even talked about it, but if you add an acid, it does help to speed up the reduction. But really, at this point, it's almost exclusively selective to the formation of CO, which is kind of great, although it sounds a bit weird to say that you're making only CO, because you only make 1 product out of CO2. And really, in my own views, the challenge with CO2 is not to reduce it in anything, it's to reduce it in 1 unique thing, whatever it is, to be selective.
So we've got a question from Dmitry Nagani. apologies if I've pronounced that wrong. So what do you think is the next? What do you think is going to be the next big development in organic electrochemistry? What techniques or approaches are going to become popular in the next few years? I guess I've got a question to follow up on that.
I don't know. I mean, really, I don't know, because I usually always have a slide about that, but made it a bit shorter this time. It's not a new technique. It has been around for like hundreds of years. So it's a bit like people start to look again at something that has always been there. I mean, to be honest, I've been doing that. I always joke, I've been doing that before it was even a thing and trendy. But then people call me hipsters for saying that. But the problem is it has been there. So I don't know what's the next development. Maybe the next development, I mean, is something that has always been there. I mean, you know, that it's a fantastic field, but people are not always good at re-saying themselves. Bioelectrochemistry is fantastic. I have colleagues in France that can use electrochemistry to detect a single molecule coming out of a cell, and you can monitor communication out of a cell. Single molecule electrochemistry, you can modify on the surface a single molecule by using electrochemistry. Spectral electrochemistry or photoelectrochemistry now is maybe the next topic when you combine, you know, electrochemistry and photochemistry. I honestly don't know. I think it will be whatever people want the next thing to be.
So in terms of industrial application, you mentioned about the flow chemistry and how you got moderate results. Was that after heavy optimization, or and
no,
if so, how do you actually go about, what is the approach with optimizing this kind of chemistry?
Okay, about the flow chemistry we've made, the results are good. It just means that it didn't really add a lot of percentage in your yield compared to the batch one, in our case at least. But I can tell you, without really saying the names, we have been developed or helped develop an industrial process. Well, not fully, but we moved from milligrams to 100 of grams with one process. It took us 1 week to move from like tiny electrolysis cell from the electrocene to the flow system, because it really depends. But when we work with the companies and they tell us we want it to go to the process at one point, we have a different mindset in the lab, because we know that we cannot use catalysts. It's possible, but in flow electrochemistry, catalysts are going to be annoying because you cannot immobilize it. We make sure that we can use technical solvent that do not require to be degassed or dried, really. And when you have a very robust system, maximum 1 week, and the only thing you have to do is just to optimize the flow. So you try several flows to see if your compound at the end is fully converted, or if you get like a clean reaction. But yeah, we have moved from milligrams to 100 of grams in 1 week.
That's really cool. Fred?
Yeah, sure. Kevin, you have a question from Jorg Blankenstein, and he's asking about the melanamides that you talked about, and he's saying, can you use something else than the carboxylic acid for R2, for the production of the R2 radical?
The answer is, why would you? But no, I mean, I don't know what you would think about. The only thing that works is that if you trap your radical, or if your double bond where the radical is going to cyclize on ends up with a tertiary radical, because you put a gem dimethyl maybe at the end of your double bond, you can over oxidize the radical into a carbocation and either induce an elimination or trap it with a solvent, methanol, to put a methoxide there. This is one thing we can do. Unfortunately, if you're thinking about using sulfonate, maybe to trap it with another radical, that doesn't work, simply because the sulfonates are actually desulfurizing at a lower potential than carboxylate. So you consume all the sulfonate before you touch the carboxylate. The other thing that works, but I don't know, you can trap it with tempo if you want to, but then I don't know what you do after with the tempo adducts. So persistent radical can trap it. That's all we have tried.
Okay, because you mentioned the alkynes the free radical from the alkynes. Have you tried
Yes.
alkynes in it?
No, I don't think. I mean, in some, I know that it would. I think actually, to be honest, in that work we tried with one. It does work. You can have an alkyne. It works. You can chop it. Again, to be overly honest, we didn't try a set of alkynes because the student told me that it was a pain to make, and she took like probably like 2 weeks. So I was like, fine, fair enough. I mean, it was probably. But
That's
yes,
true.
it does work. Yeah, it's the main reason. I mean, it's probably mostly because it took too long to make the starting material. But we've done it in the paper. There's one alkyne, and it's working.
2 weeks to make starting material sounds pretty quick, to be honest.
Yeah, no, not fast, because we are not very good at synthesis. You do not look at us when we do things. We are like, sometimes it's a miracle that we got the product.
So we got a question from Sebastian Cosgrove from Manchester. So how modular is your flow system you've designed? Can it interface with other flow methods? That's a pretty good question.
It's basically as modular as you would like it to be, because it's 3D printed and the files are online, so you can modify it the way you want. I mean, you can sandwich it with all the different cells if you want. It can flow from one cell to the other one. It's very basic. As I said, it's probably not the thing you want for production, obviously, but it's a nice little trick in the lab whenever you just want to give it a try to a flow system, because you don't want to invest, or you don't have the space or the time really to run a full, I mean, a real big, large flow system. But yeah, I mean, we are working on making with Steve like a photochemical cell to couple to that. But yeah, it's as modular as anything you can imagine, basically, because you can print it. If you can imagine and print it, you can connect it to that. We're just using HPLC tubing. It's nothing fancy.
So is it literally just the cell and then that's attached up to standard flow pumps and stuff like that, or?
It's not even pumps. We just use compressed gas to push the system.
Oh, right. Okay. Yeah.
So but you can use a pump. I mean, it works obviously with a pump too. But it's just like I always joke. I think that's a MacGyver type system. It's just a tiny cells that's very printed and it works.
That's really cool. Should we take a couple more questions? I think we should probably wrap up at that point.
Kevin, yes, you have a question from Jason Tierney.
Make it like one last question, guys.
Oh, okay. Well, Jason gets the honour then, I guess. It's just it's a follow-up really on the scale-up that you were talking with Oliver about. He's just asking what sort of quantities have been made, what sort of scale has been run with an electrochemical synthesis?
In the world, tons. If you go to BASF, they're making Lismerol, which is the scent of lily of the valley, or they make tons of it using electrochemistry, like literally. I mean, you can go and visit. I mean, I've done the visit. First of all, I mean, the smell is very strong when you get there. But they make tons of that thing using their own cell. It's a very interesting for engineers. It's fascinating the way they design it. It's not a flow cell. It's weird, but yeah, in my case, I moved from 5 milligrams to 100 grams in 1 week. That's my all-time record. But yeah, I mean, it's not too difficult to think. I mean, think about it. Compared to other methods, all we have is 2 electrodes in a batch, and all we do is just to flip them on horizontally and flow the thing through it. So there's not a big difference, to be honest. So it's one of these methods that is fairly easy to transpose to large scale if needed.
So when they do scale it up, is it just a standardized batch reactor with 2 really big, you know?
It depends.
Yeah.
It depends on the companies. It depends on the industry. It all seems to have, I don't know what the reason behind it, but they have their own preferences. I know BASF is using kind of a flow reactor, but then it goes down again, like a vertical flow reactor. It's fascinating, to be honest, but some other companies, they use gigantic electrodes, like a 1 square meter platinum electrodes. I mean, I've never seen that in my whole life, and using batch electrolysis. Some companies are using flow electrochemistry. In my own opinion, I think flow is probably the easiest to do. But they all have their own setups, and who knows why it's designed like that. But I've seen almost everything.
Yeah. I only asked because you mentioned that these reactions tend to happen at the right on the electrodes. So when you scale it up, you're obviously going to get surface area changes.
Yeah.
So
yeah. But it's not a big issue for electrochemistry because the factor we look at is not really the current. We always measure what is called the current density, the current divided by the surface area of your electrode. So you know that parameters is fairly easy. They are fairly easy to just transpose to larger scale. If you use a huge electrode, you need a very good power supply because you need to multiply the current. But ultimately, see it as being just an array of small electrodes, the same as you had in batch, but just next to each other, and that's what is happening. So that's usually not a challenge.
Cool, thanks. Really, really good talk. I think we should pass over to Nessa again.
Well,
yeah, that was a great talk. Thank you very much again, Kevin. Anyway, that was a fantastic talk, and to see more talks, then next week we have Justine Slabu from Johnson Matthey and Stephen Thomas from the University of Edinburgh. And they're going to be talking about modern approaches to reduction reactions and biocatalyzed nitro-reductions and main group catalysis for hydroboration reactions. The week after, we have some medicinal chemistry. This is from Scott Bagley at Pfizer in the US. He will be talking about palladium catalyzed preparation of cyclic alkenyl sulfonyl fluorides, a new motif for sulfur fluoride exchange, and Richard Horan from GSK, who will be talking about control in process development, evolution of the manufacturing route to versificants. Well, thank you very much, everybody, for joining our webinar. Thanks once again to Kevin for being an excellent speaker, and to my co-presenters, Ollie and Fred. Thank you.
Thank you.
Thanks, guys.
Yes.
That was really
Bye,
good fun.
everyone.
Bye.